following is a presentation of a system for online monitoring of diaphragm EMG and respiratory mechanics. The system can be used to determine the effort of the inspiratory muscles and provides early indications about the development of diaphragm fatigue. The system is designed for convenient evaluation of patients with pulmonary and neuromuscular disorders, as well as in patients being weaned from mechanical ventilation in the ICU. The project is a collaboration between McGill University, University of Montreal, and Inspiraplex. An esophageal catheter was designed in order to measure the EMG of the diaphragm and esophageal and gastric pressures simultaneously. The catheter is made of silicone tubing for patient comfort and is shielded in order to avoid electrical disturbances in the EMG signal. At the distal end of the catheter, a latex balloon is mounted for gastric pressure measurements. Proximal to the gastric balloon, 9 rings are arranged 10 millimeters apart to form an array of 8 consecutive bipolar EMG electrode pairs. Esophageal pressure is obtained by a second latex balloon positioned proximally to the EMG array. We have constructed an EMG amplifier which consists of 2 units. The first unit is a miniaturized 16-channel differential EMG amplifier which can be placed on a lever and positioned close to the patient. This will reduce noise disturbances. The second unit provides the signal bandpass filtering, isolation power supplies, and isolation amplifiers for protection of the patient against leakage currents. Separate transducers for measurements of flow, mouth pressure, esophageal pressure, and gastric pressure are also placed close to the EMG amplifier on the lever. The amplified EMG signals are analog to digitally converted and acquired into a personal computer at a sampling rate of 2000 Hz per channel. Flow, pressure, and other mechanical signals are converted by a separate A to D board and are acquired at 100 Hz. For patient protection, all electrical units are connected to the mains via a super isolation transformer. The proximal end of the catheter is easily connected to EMG amplifiers and pressure transducers which are placed on the lever. The connector which carries the EMG signals is attached to the EMG amplifier and the shield is grounded. The Teflon catheters which carry the pressures are connected to the pressure transducers for esophageal and gastric pressure. Mouth pressure is fed into the third pressure transducer. The catheter is passed through the nose and positioned such that the EMG array is at the level of the esophageal hiatus, allowing for recording of crural diaphragm EMG. In order to minimize disturbances in the EMG and pressure signals, the EMG amplifiers and pressure transducers are positioned close to the patient. In order to obtain correct pressure measurements, the volume of the gastric and esophageal pressure balloons is manually adjusted with a syringe. Calibration of the mechanical parameters is performed in a separate program where each parameter is calibrated separately. The calibration for each channel is performed in 2 simple steps. For example, in the calibration of flow, which is integrated to volume, the first step is to input a known volume into the system at a relatively constant flow as a reference. Second, a zero flow sample is taken. For pressure signals, first, a signal for a known pressure is input into the system. Second, a zero mouth pressure is selected. Maximal transdiaphragmatic pressures, here performed as a sniff maneuver, can be obtained with feedback to the subject. This module displays transdiaphragmatic pressure in red, esophageal pressure in green, and gastric pressure in yellow. Following a maneuver, the highest transdiaphragmatic pressure is recorded and displayed by a line to the subject as a target. The final accepted value is automatically added to the already existing calibration file. The system uses the ECG signal to select the diaphragm EMG signals and to calculate the heart rate. With the esophageal electrode in position at the gastroesophageal junction, ECG signals obtained from all 8 electrode pairs are of the same polarity. When signals from all electrode pairs are added, the ECGs of the same polarity will add, whereas the diaphragm EMG signals will cancel each other as they are reversed in polarity above and below the diaphragm. A strong ECG signal can therefore be obtained by adding the signals from all 8 electrode pairs. ECGs are eliminated from the diaphragm EMG signal by continuously selecting each EMG sample between 2 consecutive R waves. 2 adjustable cursors can be positioned on the screen to indicate the user-defined segment of the RR interval from which to select signals. Between each ECG RR interval, all processed EMG signals are displayed. Center frequency and RMS values are displayed on the right side of the screen. The position of the diaphragm along the electrode array is determined by systematically cross-correlating EMG signals from every second pair of electrodes. The cross-correlation technique can be successfully used because the EMG signals obtained below the diaphragm are reversed in polarity from the signals obtained above the diaphragm and hence will correlate negatively. The most negative correlation coefficient represents the region where the diaphragm is located along the electrode array. The blue signals demonstrate the 2 most negatively correlated signals in this example. This technique ensures that the EMG array and the gastric and esophageal balloons are in appropriate positions. Once the 2 most negatively correlated signals are determined, indicated by blue or green signals, a new EMG signal is created by subtracting these 2. The double subtracted signal, displayed in green at the bottom of the screen, is only shown for samples of acceptable quality. The double subtracted signal is free of electrode filtering and improved in signal-to-noise ratio. The double subtraction technique results in an increased amount of signals of acceptable quality by more than 30%. Signals which are yellow and red represent those that are too filtered by distance to the muscle or are not of acceptable quality. All 8 EMG signals and the double subtracted signal are conditioned and converted into the frequency domain by a fast Fourier transform. An average power spectrum representative of 1 second of EMG is displayed for the double subtracted signal. A reference power spectrum in green can be selected and used for comparison to incoming spectrums. Center frequency and RMS values for the average power spectrum are displayed, as well as the channels which indicate where the diaphragm is located on the electrode array. 5 indices which are used to evaluate the quality of the EMG power spectrum are also displayed. Signals are included in the analysis only if they have acceptable signal-to-noise ratios, signal-to-motion artifact ratios, and if their power spectrums are not deformed. Input keys are available for manual adjustment of the transdiaphragmatic pressure, correlation coefficient levels for detection of diaphragm position along the array, breath detection levels for determination of start and end of inspiration, and EMG signal quality indices, such as the signal-to-noise ratio, the signal-to-motion artifact ratio, the drop-in power ratio, the skewness index, and the power spectrum shape index. There are also various functions available to eliminate motion artifacts and to reduce noise. For each breath, the system detects the start and end of inspiratory flow, which makes it possible to calculate the parameters which are relevant for the analysis of respiratory mechanics online. Here, for example, is a breath-by-breath display of flow volume curves. Inspiration is plotted in red and expiration in yellow. Here is an example of a Macklem diagram, with esophageal pressure plotted on the x-axis and gastric pressure plotted on the y-axis. Again, inspiration is plotted in red and expiration in yellow. From the mechanical parameters recorded, the system also calculates fatigue-related parameters such as the diaphragm tension time index. In this example, the duty cycle is plotted on the y-axis against the relative transdiaphragmatic pressure plotted on the x-axis. Simultaneous display of center frequency and RMS from the double subtracted signal and the position of the diaphragm along the array are also displayed. The screen which is displayed demonstrates center frequency normalized to the value obtained during resting breathing here in the red upper trace. Transdiaphragmatic pressure, expressed as a percent of the maximal voluntary transdiaphragmatic pressure, is presented in the lower trace in white. These values were obtained in this subject breathing for 7 minutes at rest, and then with the progressively increasing inspiratory resistance. Center frequency of the EMG power spectrum reflects the action potential conduction velocity along the muscle fiber membrane and is known to decrease with increasing loads, which eventually lead to task failure. By following the diaphragm EMG center frequency, one can obtain early indications about diaphragm failure. On the right side of the screen, in numbers, are the simultaneous values of absolute center frequency, RMS, and the position of the diaphragm along the array. Signal quality is provided for every EMG sample obtained. We would now like to demonstrate the dynamics of the EMG center frequency and relative transdiaphragmatic pressure as measured with our system in response to an inspiratory load which is often encountered in patients, for example, during weaning from mechanical ventilation. First, we asked our volunteer healthy subject to breathe at rest for 5 minutes in order to obtain a baseline. Then, we asked the subject to breathe at 30% of the maximal transdiaphragmatic pressure against a resistance for 3 minutes at a duty cycle of 0.5. This type of resistive breathing is known to produce diaphragm task failure within 45 minutes. Note the progressive decline in center frequency after only a few breaths of the increased load. We have now jumped 2 minutes forward in time, where the subject is still breathing at 30% of maximal transdiaphragmatic pressure. It is clear that the center frequency has now reached a plateau at significantly lower values, and that the dispersion of the center frequency values remains low, as indicated by the red upper trace. We have now jumped forward in time and have obtained a recovery period of 10 minutes after the cessation of resistive breathing. From the point where the resistive breathing was discontinued, the transdiaphragmatic pressure returned to 5% of maximal, and the center frequency started to progressively return towards the baseline value. In this presentation, we have tried to demonstrate that the acquisition, analysis, and display of diaphragm EMG and respiratory mechanics can now be performed objectively, reliably, and online by a fully automatized digital system. The system has a potential diagnostic value for patients being weaned from mechanical ventilators or for patients with other respiratory muscle-related disabilities.